Saklain Sieda, High Commissioner of the Islamic Republic of Pakistan in Nairobi, Professor Margaret Hutchinson, the ABC Research, Innovation and Enterprise, Professor Leonida Kerubo, the Dean, Faculty of Science and Technology, our esteemed partner, Professor Iqbal Chaudhry, Coordinating General, Comstech and Director, ICCBS, Professor Paul Denny, Durham University, UK, Professor Batira Rossi, I'll not try and pronounce the university, Brazil, I know I need to get around it. Professor Carlos Robelo, again, University de la République, Professor, I think, yeah, all. So all our participants and those online on our platform, I really wish to welcome you all to the University of Nairobi. And I say, as we do it in Kenya, karibuni sana. Welcome. So please enjoy the Kenyan hospitality and fellowship, and also enjoy our weather. It's very nice and warm. I know in many other places, it's very hot. Now, the international workshop, whose topic is identification of new lead compounds and diagnostic tools for Leishmaniasis, concepts, approaches, capacity building, is timely. Why? The world is undergoing climate change, and there is substantial impact on our environment and ecosystem. And it is altering the way we humans live and impact on our environment and how we utilize our resources. There is increased risk of zoonotic infection and diseases. Yes. So as the global temperatures continue to rise, the impact on public health, migration, food security, land conflicts, and as new environments become favorable, exposure to diseases, disease-carrying vectors. Now, I'm aware that Leishmaniasis affects over 400 persons. I know I'm being conservative in the world, and it affects parts of Africa, Asia, and South America. And the population targets usually that are affected are communities on the edge. So usually they are affected by poverty. They don't have sufficient food and on a daily basis, and that makes them very vulnerable. So it's notable that Kenya is in Africa. So we have pockets of Leishmaniasis cases. Now, participants, the disease presents itself in a complicated manner. It is not just the parasite which leads to the disease. We know that very well. If you just have, for example, tuberculosis virus, you get the disease, and that's it. For Leishmaniasis, it's really complicated, and it incubates itself in very many organisms. Of course, man being very affected, but you also have animals. We have the vectors that carry the parasites, and you also have the environment where it sits. For example, the eggs and whatever will sit there in the environment. So there are so many factors. So some people are also asymptomatic. So you can't have the disease, but I mean, you have the parasite, but you don't have the disease. Yet there are those who are affected drastically by the disease. So the disease is not only affecting our health, but our well-being. We are no longer so confident, and it's also affecting our livelihood. Yet, it is a neglected disease, meaning it's forgotten, it is hidden. So we take cognizance that this neglected disease will continue to affect our communities, and that the effects will become more acute with climate change and the emergence of new diseases like we've had COVID, and there are studies that have shown how COVID has affected Leishmaniasis and the case. So we cannot hide, we cannot forget. The world is a global village. So we are all affected, no matter where we are. Distance is no longer significant. So this workshop comes at an appropriate time. It aims to provide appropriate diagnostic techniques and management of the disease. And this is very encouraging and appreciated. Thank the organizer for this. And the use of blended formats is also very beneficial, because it means that more people can get access to hands-on knowledge on the management of the disease. Now, this is in line with the policy of the University of Nairobi, and our aim is to have solutions for mankind, for the myriad of problems that are occurring within the world. So members, thank you, and I welcome you once again to this international workshop, and I wish you all fruitful discussion. And I really look forward to the tangible output at the end of the workshop. So karibuni. Okay, so I would now like to call upon our partners to please come and give their statement. I would like to kindly begin with Professor Dane.
Good morning. Bon matin, salam alaikum, jabbo. Use your language as you wish. Don't expect me to speak any other than English, I'm afraid. But there we go. So many thanks for that very detailed introduction. There's a few points I want to make. I haven't scripted anything. I have a thought about this, but I just want to sort of give an understanding of where we come from as Europeans coming here to Africa to deliver this kind of workshop. So firstly, I think for all of us who've traveled, be it from South America or from Europe or from Pakistan, it's a great privilege for us to be here with all of you from across different nations in Africa, and a great privilege to be here in Nairobi, as I'll talk about a little bit later. So the first thing I should say is the organization of this has taken a lot of effort from a number of people here in Nairobi, also in Karachi as well. So a particular thanks to Professor Sama Youssef, who's sitting at the back. Give her a wave. And to Professor Solomon Darisa, who cannot give a wave because he's holding a camera. But they've both worked together with me to put this together over the last probably 4 or 5 months, really, since we've started working on this. So it's great to see you here. Now, thinking about the disease, Leishmaniasis, as we were told, it's a neglected disease, a neglected tropical disease. Now, it's very clear to me that visceral Leishmaniasis, which I guess some of you are familiar with, maybe from your nations, maybe from your family and friends, is a problem, particularly in East Africa, but it's a well-recognized problem. Cutaneous Leishmaniasis is what I would style as a neglected disease. So I believe we have 1 or 2 delegates from Ethiopia. Hello, yes, we've met. So cutaneous Leishmaniasis in Ethiopia is poorly reported, and it certainly wasn't reportable by law when I last had communication with people who were working there. So it's much more prevalent in rural parts of Ethiopia than maybe I realize, and maybe than you realize, I don't know. But it's one of those diseases that is pervasive across large parts of the world, including large parts of Africa, without much reporting, with a lot of shame, embarrassment, social exclusion, and economic hardship, as well as medical pain and discomfort. So I think we need to recognize that. So whilst the workshop has some very, very generic skills around the topic of leishmania and drug discovery, which are applicable to other diseases as well, we're also going to have my colleague Emma Cole talking about diagnostics for leishmania, which is another very important point, because particularly with cutaneous disease, we lack good diagnostics, molecular diagnostics, in addition to lacking any vaccines and any real appropriate drug therapy as well. So when we move forward from this workshop, and when we move forward as a community working on leishmaniasis, it's not really me that's going to be driving the work, it's you. Because you're the people who are, well, you're younger than me, for one thing, most of you. I'm just looking at Carlos there, who isn't, but most of you, from the second row onwards, are younger than me. But also, you're the people in endemic countries. And one thing I've noticed in terms of the way funders are viewing things now, compared with maybe 10 years ago, there's much more of a drive for us to work with you, to develop you in your countries, rather than us to take the best talent and remove it and work with you in our countries. So I think there are opportunities for many of you to work in your own nation, which is what I know that most of you want to do. You want to help your nations develop and cope with the problems of those countries. So we are here to help you. We are here to support you through this workshop, and we're looking forward to talking to you throughout the next 4 days. So thank you very much, and I look forward to speaking to you later.
Thank you, Professor Dennis, for those comments. I would now like to invite Professor Iqbal Chaudhry to make his comment. Thank you.
Good morning, Assalamu alaikum, ladies and gentlemen, girls and boys, members of the Dice Party, Excellency, the High Commissioner of Pakistan. She is a wonderful lady, one of the most senior diplomat, but her humility and her excellence is reflected in everything, and we are very, very proud that she is representing our country in Kenya and of course many other countries in this region. So she is indeed a very important part of our science diplomacy effort. Dear friend, Ms. Paul Denny, thank you very much for being such a source of inspiration and strength for us. He has been motivating the entire team of GCRF NTD, and we all feel extremely happy to participate in many of these activities. We have our dear friend, the Dean, Faculty of Science and Technology, University of Nairobi, Professor Kiribo. She is the lady par excellence with lots of commitment and energy. We have the entire team of Durham University and our partners in GCRF Entity Network. We have Patrick Steele here. We have Carlos from Uruguay. We have Vatira from Brazil, and we have MK. Many more. They all traveled long distances. In fact, the people who came from South America have been, Carlos said that he was in four continents in 24 hours. He was in South America, then he was in Europe, then he was in Doha, Asia, then he came to Africa. So it's really a global trip for many of them. See, we have been part of this very, very interesting project since last several years, and we have learned a lot. We have developed a very strong network of people who are passionate and committed to work in this field. And we are equally happy that we continue to work towards that. Even that project has ended, but during our last visit to Durham University, when I summer was visiting there, we have discussed how to continue this mission. And as Paul said, that the network was initially for 6 countries, but of course we need to expand it to endemic countries in Africa. Is the right place to organize this capacity building event. So our motivation was to expand the network for future collaboration, to identify young people who have interest in this field. And we also fully understand that your impact and the impact of your work in your country is going to be immense, and therefore we need to bring you on board and whatever we can do. It was also discussed when we had a meeting with the Deputy Vice Chancellor of Durham University, Professor O'Malley, that we would expand the activities and include women scientists from Africa. And then, of course, we would actually focus on more adoptable and affordable technology. See, I've been working with Africa since last 20 years as my capacity as the director of one of the largest research centers in Asia, which is called International Center for Chemical and Biological Sciences. And I was able to bring over 1,100 African young people to Karachi, over 1,100. Even now we have scholars from Sudan, Uganda, Nigeria, Cameroon, and also from Egypt in Karachi. So we work very closely with African countries. And one thing which I would like to tell you that I've been always extremely impressed from their commitment, their capacity, and their hard work. And I'm absolutely delighted that we continue to have this collaboration with you on the platform of ICCBS and comstech. Now, a few words about comstech. Comstech is a ministerial standing committee for science and technology, 57 ministers of 57 countries in Organization of Islamic Cooperation. This is the second largest political group after United Nations, a very powerful, very effective alliance of countries in 4 continents, from South America all the way to Asia. So, we have two member states in South America. We have 27 member states in Africa, all North Africa, West Africa, East Africa, part of Central Africa, Sahel region. They are member states of comstech, and then of course we have West Asian, Middle Eastern, South Asian countries. And comstech is one of the most active organizations in the OIC system. We have the largest scholarship program. 700 fellowships of something like $2 million are given every year for the mobility of young people, largely from Africa, to various OIC countries. And we have a very large Health Africa program. So I'll tell you that we were in last 2 years, we were in Niger, we were in Chad, we were in Khartoum, Sudan, And now this year, we're planning to be in Burkina Faso. We are planning to be in Senegal, arranging health camps, arranging capacity building, training of doctors in the field of technology, cancer, and neurocritical care, and many more. And this is one of the ongoing programs. Along with this, we also have a very large biomedical training program for technicians and students. And of course, many research fellowship. Comstech is headquartered in Pakistan, so we receive lots of generous support from Pakistan. Pakistan is contributing about $3 million, and of course, then we get contribution from the other countries also. Despite all of it, Comstech, with such a mandate of science, technology, higher education, environment, and health, has a large canvas of activities, and of course, resources are very constrained. And that is the reason that we are working closely with many countries. So for Comstech, we don't really distinguish between Muslim countries, OIC countries. We work with everyone because science has no religion and science for everyone. So we work with the National Foundation of Science in Sweden. We have a very large program with the International Center for Genetic Engineering and Biotechnology in Trieste. We work with UNESCO. We work with the World Academy of Sciences. And of course, very effective program now with NTD, through which we are organizing lots of webinars, and we continue to do that in future, also finding ways and means of focusing on various of these events. Let me also add on what Paul said. I would like to thank the wonderful team of Durham University. They are global citizens, truly and honestly, they think of the entire world. And I have been always very inspired and motivated whenever I meet them. They truly and honestly believe on global human capacity. They believe on the well-being of all the people on the face of the earth, and they try to do whatever they can to help people. So I'm very impressed and absolutely delighted and grateful to them. Summer, thank you very much for being such a motivation for all of us, and of course, your very hard work from ICCBS. My colleague Fatima, I don't know whether she's online, but she has worked in the organization of this event from the comstech side. She wasn't able to come, but I'm sure that her hard work of bringing people from different countries. Right now, we have 17 countries represented. So truly an international event. We're planning to have next event. We need to figure out what would be the modus operandi where the finances would come, but we intend to have this capacity building efforts continue. And let me right at the end like to express my gratitude to the wonderful team of the University of Nairobi. You know, this university is absolutely wonderful. We have identified you, the dean, your university, as the destination of many of our future activities. This is because we are forthcoming and hardworking staff. And if I must really name one person, Solomon, thank you very much for everything which you have done. Thank you.
Thank you. I think we need some big clap for that. Okay, so I'll now follow the program, and I would like to invite Professor Bartiro. Please welcome.
So good morning, everybody. First, I'd like to thank all the organizers, particularly Paul Denny, for inviting me to come to this workshop. Actually, he said that I invited myself, but I really wanted to come to Kenya. And so with Paul and Sama and Solomon for the warm reception. Well, I come from a country that is endemic for all forms of leishmaniasis, cutaneous and visceral, and then also the subforms of the disease. So actually, I think it's the only country in the world that has all forms of leishmaniasis. And although our government in Brazil provide all treatment for leishmaniasis for free, still the number of cases never go down. So why is that? So that's because we need better drugs. We need drugs that are more effective, less toxic, cheaper, and that can be easily applied by the patient. And initiatives like the GCRF that has been funding many projects on leishmaniasis drug discovery has been very important to help finding these so needed drugs. And that's why we are here in Africa, and many countries in Africa are endemic for leishmaniasis. And this workshop, I hope, will provide new insights in the drug discovery, not only for leishmaniasis, but there are other parasitic diseases here that can, all the knowledge you can have from the presentations, I'm sure that can be applied to other diseases, not only leishmaniasis. So I hope that you enjoy this workshop, and thank you very much for being here. Thank you.
Yeah, thank you very much for your comment. And I see we are really going to work hard. We are very enthusiastic. The team is enthusiastic. So please, students, be ready. So kindly, I would like to invite Professor Robelo to also give his comment.
Good morning, and thank you very much for the invitation. Thank you to the University of Nairobi, to the University of Durham, to the ICCBS comstech. It's a pleasure to be here after being in the continents before Africa. I have in 4 continents in 24 hours. And I think we will enjoy this meeting with people with different knowledge and information. The key words of the meeting, is Leishmaniasis, and maybe we can add another keywords like neglected. Or neglected means not only neglected diseases, but also neglected situations, countries, and social and economic conditions. And that is important because both the biology and the social situation are together. One thing. And the other thing is that the other keyword can be mutual aid or networking. This started, I met Patrick and a group of girls, also are here from Brazil, near from you, and also Sandra and Iqbal, to a network, which is the network, and there we started to think in another possibility. And I think that this is fantastic because probably it will generate new networks, not only between professors, but it's important for the students, for the young people, that you will create the networks of young people that will be the future network. And I come back with networking and mutual aid. That is a form of make policy to the mutual aid between countries, the different countries, situations. So I am almost sure that you will create interaction. Now it's easier with WhatsApp, internet, etc. That will be for the future very important. Thank you very much, Solomon, Summer, members of the University of Nairobi, and all the people here. So we will enjoy this. Thank you very much.
Thank you very much. You know, we listened to South American, like I told you, I always remember football. Such a pleasure to hear from you. Now, a very humble request to make to Professor Iqbal Chaudhry to kindly come and invite our invited guest.
Ladies and gentlemen, boys and girls, you see, there are always some people who really inspire you to the core of your heart. And our ambassador, Her Excellency Saflan Saida, is one of those. I met her in Kenya during the last event, and we spent some very good time together, a lot of very insightful discussion of bringing Pakistan and Kenya and other countries together. Our commitment, our excellence, our motivation is just exemplary. As I mentioned that in Ministry of Foreign Affairs, the Foreign Services, they are among the best people in terms of in capacity to lead, and she is among the best of the best. So with great pride, I invite Her Excellency, the High Commissioner of Pakistan, and present to you. Thank you so much.
Bismillahirrahmanirrahim. Assalamu alaikum. And thank you very much, all the learned, honorable guests here and seated here with me also. I did sciences up to my O levels, and after that I didn't do so. I'm not so well versed in science, but in my 3 decades of diplomatic service, you know, I have actually lived on all the continents now, except Latin America. So you can say that I have been exposed, have understood various diseases that I came across. My first endemic was H1N1 in East Europe when I was in Ukraine. So we had a lockdown, and this was my second lockdown with COVID. Then in the last 2 decades, we have seen diseases like from green monkey disease in Africa to SARS and then to COVID. And like for dengue, at that time, we didn't have it in Pakistan, but I was exposed to it when I was posted in Far East. So I really had dengue also. So this thing is very informative also. And when I read the name of this disease, I hope I pronounced it right, Leishmaniasis. It is something that is very new to me, but it gives me an understanding of how isolated we all are. And this is one of those steps that will actually bring us together. And COVID exposed that isolation, you know, very well. We did not know what to do, how to do, and how to engage with people. But coming to those diseases which are already there, that was a new one. And that made us realize that, you know, we really need to work together. And diseases like this which have been there for a while, and where various countries like Brazil and others have been working, you know, on their own. And even in Africa, like Ethiopia, we need to work and we need to bring it out in the open so that at least people are aware of it, number one. Number two, if God forbid, since as our host mentioned that distance is imperative, is a relative term now, you know, like it doesn't exist for us. If anything moves from one place to another, like for example, dengue, it just came in a little container to Pakistan, and now it is a very lethal and a fatal disease. So we should be all prepared, and we should have an understanding rather than, you know, hitting the darts in the dark and find out what the reason is and why we are having it. We should really take it by the horns. And thank you, Dr. Iqbal, the Durham University, and Dr. over here, so for bringing us together in this platform in University of Nairobi, who's hosting us, and for increasing the collaboration which is not only a south to south engagement, but also a north to south engagement, a global engagement. Thank you very much.
Thank you very much, Your Excellency, for putting us on the floor. Thank you. Now, I would like to invite the ABC, that's the Associate Vice Chancellor, Professor Margaret Hutchinson, who is here represented by our able Dean, the Dean of the Faculty of Science, Professor Leishmaniasis.
So thank you very much, Catherine, for that good invitation. May I acknowledge the presence of the following, Your Excellency, the High Commissioner of the Islamic Republic of Pakistan, Mrs. Seklain Saida, Professor Dr. Iqbal Chaudhry from the Coordinating General of comstech and Director of ICCBS, distinguished resource peoples from Durban University, Professor Paul Denny, Professor Patrick Steele, and Professor M.K. Paul, and also including Professor Batira Rossi, and Professor Dr. Carlos Robelo from the Universidad de la República Uruguay, Professor Samia Yusuf from ICCBS, University of Karachi, our international and local workshop participants, ladies and gentlemen, good morning.
Good morning.
It is with great pleasure that I welcome you all first to the Faculty of Science and Technology in my own capacity as the Dean of the Faculty, and also to the University of Nairobi as a representative of the Associate Vice Chancellor, Research, Innovation, and Enterprise. Just briefly to talk about the faculty before I read through the speech of the Associate Vice Chancellor. The Faculty of Science and Technology is the most beautiful faculty of the University of Nairobi. You will attest to that when you go for lunch today. You can walk around, you see it is climate change, adapted to climate change, because it has trees all over, and you can see it has very old buildings, but of course, those buildings remind us of where we came from, but they are beautiful as much as they may be old. So, and I am just, the visitors, you can pay a courtesy call to the office of the Dean. I'll be very happy so that we can remember that you came there. And we have faculty members around 283, and the student population, international and also local, are around 5,000. And our researchers carry out cutting-edge research, of course, with other institutions outside. We don't do research alone. We collaborate with other institutions in many areas. When it comes to natural products, we are the lead in the region. It goes without saying. I have my mentors here. We have Professor Abi Yenesu, Solomon Jerese, then we have also students, although we've not really ventured into leishmaniasis. I think maybe because of the assay systems which are available. I think that could be one of the reasons. But we have a lot of compounds in our bank, which we've isolated over the years. So I think in this area, if we have partners to do the assay systems for this leishmaniasis, then we can do immediately. I forgot to introduce myself. I am also a chemist, and I'm a natural product chemist. So this area, I'm very conversant, only that I might not have enough time to review throughout because of the administrative responsibility. But I also wanted to our visitors and the students, and both students and also the other members, we are very open to collaboration. We can collaborate in very many areas. For example, coming up with programs, joint degree programs, dual degree programs, also in writing joint proposals. Like, for example, nowadays there is the Horizon proposal, so we are open to collaborate in that way. So, and I'm again encouraging you to visit my office and also to visit the website to see the departments in the faculty. There are 7, and also we have 3 research centers. So you can visit. I don't want to talk too much. So let me go now to the speech of the AVC, the Associate Vice Chancellor. So first of all, I thank all of you for sparing time off your busy schedules to attend this important workshop on Leishmaniasis. And with the title, Identification of New Lead Compounds and Diagnostic Tools for Leishmaniasis, Concepts, Approaches, and Capacity Building. The Office of the Associate Vice Chancellor, Research, Innovation, and Enterprise, University of Nairobi, continues to spearhead the overseeing and coordination of research programs and extension of knowledge uptake in all the faculties of the University of Nairobi. And the Office of the Associate Vice Chancellor coordinates research, innovation. Okay, I think this is a repetition. So I am aware that comstech and ICCBS on the 2nd of January 2022 signed a memorandum of understanding to collaborate on areas of mutual interest. It was during the NAPRECA Comstech Summer School last year. You recall Professor Iqbal Chaudhry had the idea of hosting this workshop here at the University of Nairobi, and particularly at the Faculty of Science and Technology. We are glad our MOU is bearing some fruits, and we will continue to work with you on many other areas of mutual interest. We have vibrant research teams undertaking cutting-edge multidisciplinary and multi-institutional research, most of which are geared towards achieving the Big 4 agenda, other national and regional priorities, the Sustainable Development Goals, the National Vision 2030, amongst other priorities. We welcome any new mutual collaborations in joint degree, dual degree programs, joint proposal writing, just to name some of the avenues of partnership. And we encourage you to visit our website to see the existing research teams. I am pleased to note that this workshop brings together top-notch researchers from Brazil. Those from Brazil, maybe I'm not really sure who came from Brazil. Maybe you can raise your hands and we clap for you. From Brazil, anyone who came from Brazil?
Yeah.
Yeah, so Pakistan. Then we have United Kingdom and Uruguay. I don't know how to in the area of neglected tropical diseases as resource persons in this workshop. This workshop has attracted senior and early career researchers as participants from Benin. Can you raise your hands? Benin, any? Oh, okay, maybe they didn't make it. Burkina Faso, Cameroon, welcome. Egypt, they make it. Ethiopia, Ghana, Madagascar, Niger, Nigeria, Sudan, Tanzania, Zimbabwe, Uganda, and Kenya. Any other persons I forgot? Any other country? Okay, so thank you very much. Welcome all of you. From the conference program, I can see the main focus of the workshop is on Leishmaniasis. As you may be aware, Leishmaniasis is one of the major diseases affecting our society. Over 5 million people in Kenya are considered at risk of exposure to the disease, with an incidence rate of 2.96 per 10,000 people affected with visceral Leishmaniasis. We are grateful that you chose our institution to host this important workshop, which is aimed at identification of new lead compounds and diagnostics tools for Leishmaniasis, concepts, approaches, and capacity building. Ladies and gentlemen, invited guests, renowned resource persons, and workshop participants, as a university, we have the responsibility and mandate to come up with affordable solutions to address the myriad of challenges our society faces, including coming up with interventions for Leishmaniasis. Let's take the advantage of academic freedom, of creative thoughts and expression to contribute vibrantly to the challenges of our times, mindful that what legacy we are imprinting. I wish you all fruitful deliberations, and we will continue supporting you in whichever way possible for the objectives of this workshop to be realized. So with these few remarks, I am pleased to declare the International Workshop on Identification of New Lead Compounds and Diagnostic Tools for Leishmaniasis, Concepts, Approaches, and Capacity Building officially open. So thank you very much. I will see you in Chiromo campus. My campus is called Chiromo campus. Thank you.
Thank you very much. ABC represented so ably by Professor Kerubo, our Dean, Faculty of Science. I'd like us to give another round of applause to all the speakers. So we've reached that point where we are now ready to, I don't know if maybe I could give one comment, if anybody would like to make a comment from the audience. Any comments? We are very democratic.
Yeah.
I'm sure that, yeah. Participants, students, any comments? Okay, I think we are all satisfied. We've listened to all the speeches and we are present. So thank you very much. Now, I'd like to ask maybe to stand up and be able to take the photo. I don't know where we'll take the picture.
Organized. Yeah.
Maybe where we have our flyer. Together about 24 countries.
Great pleasure, was involved in the first lecture of the group. Or you can move around if you wish to.
Well, we have the microphone. That's this one, we can bring it to the product. Oh, that'd be good. Yeah. So then that's what we want.
And I'll say more in fact. Because
Okay. Fine.
and if we have any issue with the visibility, kindly move to the left side.
Excuse me, sir, can you hear me? Dr. Solomon?
Right, so I'm going to have to speak in the microphone, of course, because we gladly have a number of people joining us via Zoom. Hope you enjoyed the tea break. I'm glad I got the chance to speak a little bit to some of you. I hope to speak to all of you at length before the end of Friday. Okay. So this is a workshop. So whilst this is a bit of a lecture, please feel free to interrupt me to ask a question, tell me what your view is, or tell me where I'm wrong. I'm very, very open to that. So Durham is, as you know probably by now, is in the northeast of England, and of course we are now here in. So just want to put this up to emphasise that Durham is a cathedral city, and very different sort of place to the nations that we work with in Asia and South America, and now here in Africa. But still we find common ground and we work together. So just want to take the opportunity of being the first selected speaker, which is a delightful position to be in, because I can say what I like without contradicting anyone else. So it's up to the people later to contradict me. So I want to talk about discovery of anti-leishmanials, but I also want to emphasize it's a problematic process. I know a lot of you, if not all of you, are interested in natural products. I will talk about natural products a little bit here, but I will also talk about them in a more practical sense on Friday, but Thursday, when we talk about the screening process. Okay, my second talk. So I work within the Department of Biosciences and the Centre for Global Infectious Disease at Durham. And I have long-standing interest in Leishmaniasis, which you can see here, and also in apoplecsin disease, particularly toxoplasma, which I will mention very briefly at the start. So this is our network, as Iqbal particularly mentioned, and Carlos as well. It's a network which triangulates 3 continents, and whilst the funding is finished, we still work towards the discovery of new anti-leishmanial drugs, essentially. Okay, so just want to give you an introduction to sphingolipids. I know a lot of you are chemists, some of you are biochemists. Most of you have probably heard of sphingolipids, not all of you will know what they are. So, but they're plasma membrane lipids, which are in all eukaryotes, including us. And they've been implicated in almost every function that a eukaryotic cell does. Whether that's correct or not, that is up to you to work out. But lipid rafts, for example, very famous micro domains in plasma membranes, functional units, controversial but associated with sphingolipids. Okay, so I'm not going to talk about Leishmania to start with. I'm going to talk about toxoplasma, just to give you an example of some of the work we're doing and an introduction to what Emka in this case, we'll talk about later. I'll also bring in work I'm doing with Patrick and Bartira as well. So Toxoplasma like malaria. The malaria parasite Plasmodium is a apicomplexan. It's a parasite of any cell, so it's quite easy to work with. It infects maybe half the world's population, largely asymptomatically. Definitive host is the cat. Can cause very significant neurological disease and agricultural losses as well in sheep farming, which is quite important, and is related, as I mentioned, to Plasmodium. Now, and I mention this now because I'm going to come back to it a little bit later, but the eukaryotic serine palmitoyl transferase, SPT, is the first enzyme in sphingolipid biosynthesis. I'll show you the pathway in a cartoon sense a bit later. Now, in eukaryotes, including Leishmania, it has two subunits, LCB1 and LCB2. LCB means long chain base. That's not important. That's just because it was first discovered in yeast, and yeast people like to give things strange names. So that doesn't really matter. There's two subunits to this enzyme, and they've been now well characterized, including at a very high molecular level. Now, in contrast to that eukaryotic enzyme, some bacteria, not all bacteria, have an equivalent, and it's a soluble enzyme. There is one subunit which forms a dimer with itself. Okay, so this is well characterized again at a high level. Now, what we've done, and this is work with Emka as well, we continue working with this protein, is that we have identified a bacterial-like serine palmitoyl transferase, this homologous dimer in Toxoplasma, and that's conserved in Plasmodium and other apicomplexans. So what we've done is look for differences that maybe could be targeted by drugs. So that's the point I want to make here. Look for specific, unusual features of parasites and proteozoa that maybe we can target. And then we publish it. Okay, but let's get on to Leishmania, because that's what we're here for. We heard a number of comments, including by me in the introduction stage, about Leishmaniasis. And really, you know, as I said, it's you guys in the audience that are going to be leading the fight against Leishmaniasis in Africa into the future. So it's a disease that is been pulled out of control, and in many ways it is. Even with therapies and some diagnostics, it still never goes away. It probably will never go away, because it's a sandfly vector-borne disease. Very difficult. We don't really even understand sandfly breeding cycles properly. So vector control is almost zero. And it's endemic now in more than 90 countries. It's increasing. Climate change and deforestation increases the reach of sandflies, as long as they're infected animals, because it's zoonotic as well, most forms. And sandflies, you will get transmission to people. Okay. So globally, this is a bit out of date, and it's a DNDi, Drugs for Neglected Diseases initiative figure. The WHO now reckons globally there's about 1 billion people at risk of Leishmaniasis. So the numbers are absolutely huge. And the numbers of people infected, as I mentioned earlier, 12 million is an awful lot, but that's probably a massive underestimate, because we don't know how many people have Leishmaniasis in the world. I think in Africa, the lack of reporting is probably a bigger problem than it is in, for example, Brazil. So, you know, we need to concentrate more on Leishmaniasis. So this is something I picked up on a trip to India. So this is the Indian national newspaper. And this is a 17-year-old boy in Bihar, the southeast state of India, bordering Bangladesh, with Kala Azar, visceral Leishmaniasis caused by Leishmania donovani, being very wasted. There's a big effort to eliminate, that doesn't mean eradicate, eliminate, that means you have a very low number of cases this disease in India, Bangladesh, and Nepal. Some success at the moment, but we don't really know how much success to be on. Now, I found this, and also found just last night the Kenyan government's policy on controlling leishmaniasis in Kenya. So I encourage you all to read that, particularly if you're Kenyan. But this is the Nairobi Declaration, I think of 2021. So this is really to encourage people to work together to eliminate visceral leishmaniasis as a health problem. Okay. Now, underneath that, though, we have cutaneous disease, which is what myself, Barteria, and Patrick are particularly more interested in. So this was, and also Youssef, Sama Youssef is particularly important here because she is running a great program in trying to identify natural products which have a genuine effect on people with cutaneous leishmaniasis in Pakistan. And this is Jinnah Hospital in Karachi, and this is where, thanks to Iqbal and Sameh, I was able to visit and talk to doctors and actually meet patients. This is one family of children with cutaneous leishmaniasis. So their father had taken them to see family more towards the north of Pakistan. They'd returned and they each developed lesions. And some of these, such as here, they're not resolving. This is because it's a different species of leishmaniasis. The tools are not easily available to identify which species. So that is not responding to infection. And even when it does respond and the infection is resolved, leaves scarring. So this young girl here, I think it was only 3, is going to have severe scarring on her face, which affects life chances and her mental well-being. Okay, just to kick off, really what we're all going to talk about, I will mention the life cycle. It's like when I talk to undergraduates, I will say, if you're talking about parasitic disease, it's all about the life cycle. So I think all of us, there's at least myself, Carlos and Barteria, even Patrick regards himself now as a parasitologist who put the life cycle. So I mentioned sandflies, flagellated forms that you can see here, injected into a mammalian host. Could be a person, could be a dog, could be a chicken, could be a rat. Infecting macrophages and causing, as Barteria mentioned, Brazil has a unique status here because it has all these forms of disease, either visceral, with the lung and the liver and spleen infected, particularly mucocutaneous, mucous membranes, particularly in the nose and mouth, or very, very common throughout the world, actually, most particularly in the Middle East and towards Pakistan, but also in Africa as well as this cutaneous condition. And they're the cases that I mentioned with those children in Pakistan, the Jinnah Hospital. Okay, so I know you're all a number of you are natural product chemists. So you will recognize immediately the natural products on this list. Who can tell me which the natural products are? Shout out, please. Barteria, start me off. Which of these are natural products? Amphotericin B, there's one. So this is the one that's used in the elimination program. Give you a clue. These antimonials, which are commonly used by even injecting into lesions, are not natural products because they have heavy metals in. These are synthetic drugs here. So that's pentamidine at the bottom. That's the word.
I wish, this thing.
And on the right, that's miltefosine. Which is the only one of these that's already available. The other natural product is paramamycin. So the fact it's mycin should tell you it's an antibiotic, a natural product antibiotic. So actually that's used in combination therapy now extensively for leishmaniasis. I think particularly in Somalia, in Sudan, actually. let my Sudanese colleague correct me on that later. I can't advance the slide anymore.
Yeah, so
Yeah.
that's a problem with that, but now it should be.
Oh, there we go. Okay, so they're the drugs available, and we do use various technologies to try and find out what the mode of action of these drugs are. So I'm going to talk about some of those. I can't go into detail. I'm just going to give you an overview. So I'm quite happy to talk about the detail later. So we use things like mass spectrometry to look at the effect on the metabolism of cells in a broad way. We use genetic tools to look at the role of genes in the mode of action, the mode of proteins. And we use, as Patrick will tell you, chemical tools to try and identify these. Okay, so sphingolipids, very important, as I mentioned. Ceramide here is the primary sphingolipid. It's the base unit. And as humans, we're all human, I think. Tell me if not. We all have sphingomyelin in our plasma membranes. Okay, a very essential lipid in plasma membrane. People with deficiencies, normally genetic, in sphingolipid metabolism have a whole range of diseases, and they may not even survive through to early childhood. It is a serious issue. It's a rare genetic disorder, but it is problematic. Now, however, this rather complex looking lipid here is like sphingomyelin. It has ceramide, but rather than having this phosphocholine as a head group, it has phosphoinositol. So there's inositol sugar at the end here. And that's inositol phosphoryl ceramide. Okay, that is the primary and indeed sole sphingolipid core in Leishmania, but also in yeast and plants. So there is differences. Again, getting back to the concept is we have differences. So serine palmitoyltransferase in Leishmania, that's almost exactly the same as in humans and other mammals. But as we go towards the production of ceramide and the complex sphingolipids that accumulate in our plasma membranes and have this very important function, whilst we have sphingomyelin, which is produced by sphingomyelin synthase, yeast plants and some protozoa, including Leishmania, have inositol phosphoryl ceramide synthase, IPC synthase, producing the IPC, the primary complex sphingolipid in the plasma membrane. So there's a difference here. So in drug discovery, differences when you're looking at antimicrobials between the host and the microbial pathogen are a relatively easy start point to think about chemotherapeutic intervention. So just to give you an overview, this is the procyclic Leishmania. It doesn't normally glow in the dark. This is labeled with green fluorescent protein and fluorescent ceramide and DAPI to stain the nucleus, and the kinetoplast, which is the mitochondrial genome that gives the whole group of these kinetoplastids their group name. And then there's the flagellum going off into the plane of focus. Now, inositol phosphoryl ceramide, IPC, is produced in that Golgi apparatus. And it's a very polar cell. So it's produced in that Golgi apparatus. And through this very bright body you can see there, it's then trafficked out to the plasma membrane and then arrays itself, leading to accumulation of about 10% of the plasma membrane lipids are IPC. So it has a very important function. Now, why do we think it's a drug target at all? This is the basic pathway in a biochemical sense. And what we can see is we have phosphatidylinositol, that's a phospholipid. And the phosphoinositol part of that attaches to an activated histidine releasing diacylglycerol and DAG. Okay, diacylglycerol is a mitogen. It promotes growth of cells. It makes things grow faster. That's at least the case in mammals. On the other hand, We have the other substrate, which is ceramide, which is shown here. And just look at that primary hydroxyl group. So we have that phosphoinositol there, and then the ceramide comes in, steals that phosphoinositol, and produces inositol phosphorol ceramide, and then the enzyme is reconstituted. So that is the balance is between diacylglycerol being produced and ceramide being utilized. And ceramide is the flip side of DAG in that it's a pro-apoptotic. It's a cell death mediator. Okay, so you've got one that promotes growth and one that promotes death. So this enzyme and the equivalent in mammals sits at a balance point between life and death at cellular level. And I won't go into these, but these are all natural products. But all of these have been shown to target the fungal IPC synthase, and at least aureobacidin at the bottom has been then gone into some form of clinical trials, although it's never really progressed beyond phase 2. So it's a validated drug target in the fungi, the pathogenic fungi, which are an increasing problem, and we should all be interested in fungal disease. We are going to have a global crisis with fungi and drug resistance. Not in 20 years, not in 30 years, but probably in 5 or 10 years, it will become a serious problem, including here, but including in Europe as well. So I won't go through this in detail, but I'm now going to move on to discussing screening for drugs, okay? And this will be complemented by my sort of more interactive workshop talk on Thursday. So we have 3 primary modes of screening for new compounds. We have what I've called in vitro high throughput screening. So this is a biochemical assay. So you have a target, which is normally a protein, and you put it into what is normally a biochemical assay, and then you screen for the compounds that inhibit it. Okay? And the idea is that then those compounds can be developed into drugs that will inhibit that essential enzyme in the parasite, and that will be your solution. Now, that sounds very attractive, but there's a few things you need a protein, you need an assay, and you also need suitable substrates that you can make that assay with. So none of those are simple, it can be very expensive. But on the upside, it's possibly very simple, be very, very sensitive, and you can use it to develop compounds later on. Secondly, which is what we're going to focus on Thursday to a large extent, is phenotypic high throughput screening. So there you don't need a purified target, so it simplifies it. You're literally putting compounds against a parasite in this case and seeing whether they kill the parasite. Okay, so it's a completely naive, you're not expecting or assuming anything at the start. You're just targeting everything all at the same time. Now, of course, the problem is that could be quite insensitive because the compounds that are excluded by the membranes won't be giving a hit. So you'll miss those compounds that could be developed through the smart medicinal chemistry that you guys can do into the next drug, the Leishmaniasis. This can be expensive because parasite culture can be very expensive. Leishmania has a cost, to be honest, to grow, particularly the more complicated assays, which we'll talk about soon. And ultimately, though, you also, for development, you need to work out what the target is afterwards. And Patrick and I think Bartira will mention this to some degree later. Bartira's looking confused. And what I'm going to talk about just now is a combination of these approaches where, working with Patrick and GSK at Trescantos, we developed and then used a yeast-based assay. Okay, so this is brewer's yeast, the yeast that's used in bread making, beer making, the common, easy to manipulate yeast. Now, it's easy to manipulate genetically, it's cheap to grow, and if your enzyme, your target, can constitute a function in that yeast that you can measure, then you can use that very simply. And it combines the benefits of having a cell-based assay, because they're living cells with the context of a living cell, with a target-based approach. Now, there are downsides as well. So you need your protein to work as the enzyme in this foreign environment of the yeast. And also, it's not necessarily very easy to get a compound into yeast. They have a cell wall, as you'll know, but they also have lots of pumps that pump out drugs or any other chemical you want to throw in. So it's a pros and cons situation.
Okay, now,
There we go.
good.
Okay, without going into detail, because I think detail is for discussion sort of over a cup of coffee or tea or lunch, is the way we approach this is we had a knockout of the yeast IPC synthase. It's essential, it's called all-1, and then we support the growth of yeast by expressing that enzyme on a plasmid within the yeast cell. So these are meant to be budding yeast. This is my cartoon of what yeast look like, probably very poor. And then we add into that on a different plasmid, the Leishmania IPC synthase. So we have both. Now the Leishmania IPC synthase is controlled by a galactose promoter. So we have to add galactose, the sugar, to get expression. So we have both enzymes expressed in the system. Now, how do we then test whether Leishmania enzyme can support the growth of yeast, which is the point of this experiment? Well, we use a chemical called 5-fluororontic acid. And what that does is when it's interacting with that Ura3 protein that's produced from that plasmid, which is essential if you want to select for it in the absence of uracil. But if we add 5-fluororontic acid, it produces a toxic product which kills the yeast. So what you're doing is selecting for the loss of that plasmid. So sometimes called a plasmid purge. Okay. And then we end up with a yeast which only has the Leishmania enzyme expressed, and it's entirely dependent upon that enzyme. So I'll just get some water. Okay, so that's the basis of our assay. Then months and months of actually assay workup to get it into a format that we can use for high throughput screening. And because we're working with GSK Trescantos, this is a library of 2 million small molecule compounds. So that's an awful lot. I think this is really just to give you the idea that you don't start with 2 billion and then work with 2 billion all the way through. You go through a process of triage, which you'll all be familiar with in your lab work. Some things you just ditch early because you can't work on everything all the time. So we go from 2 million to 20,000, and you end up with 4,000 hits that are selective. And when I've been selective, so we want to get rid of the non-active ones, of course. But we also want to get rid of the ones that are active against the yeast, but not the Leishmania one. And particularly, we want to get rid of those that are active against both. Okay? So we want compounds that will inhibit the yeast, which are dependent on the yeast, on the Leishmania enzyme expression, but they do not inhibit the yeast, which has its own enzyme. Now, why would you do that? Well, you do that because otherwise, we just get lots of compounds which just kill yeast. They wouldn't be target specific. So it's this stage here that's really important for making sure that we have on-target hits, which is what's green. So that's how we've gone down in this process from 4,200. This is the selectivity. So this corner here on this graph, these are all the data points from screening against the yeast, which has the Leishmania enzyme and natural yeast. And the most selective ones you can see here in the bottom right-hand corner, they are 30 or more fold more selective for the Leishmania enzyme than the yeast equivalent. Okay, this is where we identify from 2 million compounds. We've got very quickly, this is quick actually. Once you have the assay, you get down to 500 compounds. So we're trying to digest 2 million compounds. As I said, we triage very quickly. And we get down to the compounds which we can work with. Okay, so these are the ones that are active and specific. Now, another process, which you may not be familiar with, you might have heard if some of you have done some pharmacy training, is the prospect of clustering. So the chemical similarity of compounds in a compound library can be very high. And what you don't want to do is waste lots and lots of time looking at compounds which just vary in a single nitrogen or something, because they're going to be very similar. So they're clustered, and then 2 representatives are selected from that. So we go from 500 to 211. Iqbal.
Yeah, you see, from 2,500,000, what was the technology which you used for screening?
Yeah, well, I was going to mention that tomorrow, but I will mention it today in case I forget tomorrow. And in fact, you've asked a question, which I'm very grateful for. Technology is robotics. So we could never ever do that at Durham.
Yeah.
This was done by a student and a postdoc on secondment at GSK in Tres Cantos, Madrid. So these are highly complicated robots, and I'll talk tomorrow, Thursday, about the kind of assay plates used, but they will almost blow your mind about the tiny quantities of media and cells that go into each assay. So you're screening 2 million, but it is not as big a job as you think, because it's roboticized, it's miniaturized. Okay, so that's how you get around it. So automation and miniaturization. But the hard work, if you like, comes in the maybe 6 months to a year developing the assay, making sure it's robust, and then miniaturizing it. Once you put it in the robot, it's pressing the button, he says. But it is pretty much that. Yeah. Good question, though. Thank you.
Thank you.
So, okay, so then we have these 500 hits, and then this is the first time they meet the parasite. So this is several months of work, probably about 18 months. And then they meet the parasite. This is where we use amastigotes. These are Leishmania donovani, actually. So they're azenic, so they're grown without a host cell. Amastigotes are normally within macrophages. And we select those that have a high efficacy. These are log values, but we're looking for things that inhibit growth 50% at less than 10 micromolar. Okay. This is a chemical physchem measure that measures the hydrophobicity, the size, and various other factors, a number of aromatic rings, which is also here, to give a number. This is really a GSK algorithm that tells you whether you should take that compound forward, whether it's developable. And then this one, importantly, this is really relevant to all of you if you're doing screening, is the selectivity. Okay, again, a log value, but you want selectivity, and this is against HepG2 cells, these are liver cells, of more than 10, because it's a log value. Okay, so you want it to be at least 10 times more potent against the parasite than the host. So after that, we go from 211 to 25, again, very quickly. And it's only then we get something more realistic, like an infected cell model. So these cells here, they are human cancer macrophages, if I can call them that. Yeah, THP1. In green, that is a mastigote, Leishmania donovani, within those macrophages. Okay, so they're labeled with green fluorescent protein. So with a smart camera and some smart software, which we don't have, but GSK have, it was developed by Dundee, actually, Manu Duraika. You can count the number of infected cells, and you can count the number of parasites within each infected cell. You get a very global view of an in vitro infection. So from that, we've now gone from 25 to 5. 2 series, that's 2 pairs, and 1 singleton. And for various reasons we can talk about later, we focused on the benzazepines. Iqbal, another question.
I'm asking these questions on your behalf.
Ah, well, please go on.
Oh, okay. So what was the advantage of using this cancer macrophages as compared to normal macrophages?
It is the ease of growing them. I mean, cancer macrophages are not normal macrophages. It should be said, they're quite different. But it's the ease of growing them in culture. Otherwise, you're reliant on getting a mouse, particularly, and getting bone marrow. It's quite a laborious process. So by doing THP1, there's a mouse equivalent called raw 267. You get around that process, and they're still able to be colonized by the Leishmania, as you can see in that image. But it's a practical issue. It's not ideal, to be said. So, of those 5 compounds, and some and focusing on the identified benzazepines, and I won't go through the chemistry, but Patrick may cover this later. Even though we've gone through a hit identification process and some development, we still have to prove in an in vitro assay, an enzyme assay, that the compounds we identified really do inhibit the enzyme. Because yeast is still a complex cellular system, and there are other alternatives to why that compound may be a hit. And this is well, I say well recorded, this is recorded in the literature. Not necessarily recognized. So we then spent some time using a rather more complicated enzyme assay. Now, the challenge with IPC synthase is it's a 6-transmembrane enzyme. It's associated with lipids, essentially, with lipid substrates and lipid products. None of that makes a very easy enzyme assay. So we used yeast microsomes. So we extracted the enzyme in lipids from yeast. We washed it in detergent, and we actually have very active yeast extract enriched in Leishmania IPC synthase. Okay, and then I won't go into the complexity then of the assay, because that involves some ion exchange in a 96 well plate, but it works, and it's a decent assay, although very complex. You would never do it for 2 billion compounds. You probably wouldn't want to do it for more than 1,000. Very time consuming. But then we identified that the identified benzazepines, as well as some other derivatives of the benzazepines that GSK offered us, were low or sub-micromolar inhibitors of the enzyme. So that was promising. So we've gone from 2 million, and we've identified a class of compounds that does target the Leishmania IPC synthase. And then we went into the Wellcome Trust drug discovery engine. We were the first Leishmania project in this. And the idea was to move promising compounds towards preclinical and then clinical trials. And we really had to sort of get very close to the activity of, in this case, miltefosine. So we had to get close to a clinical drug. And we failed, like most projects. And I want to make it clear here, I don't celebrate failure. I do recognize it. So you will be involved, if you stay in science, you'll be involved in multiple failed projects. We all are. We shouldn't be ashamed of it. It doesn't make you bad scientists. Just recognize it, and that makes you honest people. And I'm sure you are honest people. Okay, so we published that, and we've also worked around this enzyme with tamoxifen, anti-cancer drug seems to inhibit it. Worked with Patrick, which you may well mention, clemastine, he will mention it, good, which worked with Bartera as well on the animal model with clemastine. So we see that that has gone into preclinical trials, you could almost say, but it's certainly been tested in infected mouse model and is quite effective. It's not going to be the blockbuster drug, but it's quite effective. And we even made the front cover with a nice image that they produced for us. Anyhow, but now this comes to another question, and then I'll move on to a slightly different topic. is sphingolipid biosynthesis in general really a drug target? Again, we have to be critical. We have to think about the literature. We have to think about what we know, and we have to really be prepared to change direction. So this is work from me from nearly 20 years ago, and where we could knock out, and this is why I mentioned the serine palmitoyl transferase, we could knock out serine palmitoyl transferase activity. Remember, that's the first enzyme in sphingolipid biosynthesis. So you knock that out and there is no sphingolipid. And in an animal model, we still get an infection. The infection is delayed, but we get the presence, in this case, of cutaneous lesions. Okay, this is a cutaneous disease model. And a little bit later, someone did something very similar, and still a similar observation. Okay, so this is when I sort of moved to Carlos's future talk, in a sense. And I know that most of you are probably not familiar with genomics, so I'm not going to talk about this in any detail, but we do now have the capacity to genome sequence relatively cheaply. The analysis is much more expensive because it takes skilled and experienced operatives. However, we can do it. So what we did is took the knockout that I just showed you that survived in mice, the knockout of the serine palmitoyl transferase LCB2 subunit, no sphingolipid biosynthesis. And this is just an example of a comprehensive analysis of the whole genome, all 30 plus chromosomes. And this is the wild type here, and this is the knockout. Of course, we can see that we've knocked out LCB2, but we've also knocked out in every cell something here. It's disappeared. And this is an ortholog of a transporter of lipid in humans, and so-called ABC transporter. Now, so that was missing. So then we moved into using sort of newer technology to attempt to genetically analyze this. So we moved from using, and we can talk about this afterwards, old 1980s technology, homologous recombination, and lots of drug selection, to using, I'm sure you've all heard it, the term molecular scissors, using CRISPR-Cas9, gene editing. Okay, this is going to be a big news in agriculture across the world, including here, but also it's a big tool and a big opportunity to really do drug target analysis. So when we used these molecular scissors to do the same kind of experiment, but with much less sort of time and drug selection, so a quick experiment, we could not knock out LCB2. It was essential. However, when we knocked out this ABC transporter at the same time, we recovered viable parasites. So we really have to reanalyze a lot of the knockout data, which started about 1985, 86, and really think about what other changes in the genomes happen when people like me are in the laboratory doing genetic knockouts and genetic analysis. So we need to really reevaluate lots of data. So where does that leave the IPC synthase? So what we haven't done any genetics. I tried, I failed to knock it out. So you assume that it's essential. But really, where does it leave it as a drug target when we can use these molecular scissors, this gene editing approach? So this happened years after the screen was done. So the screen was completed in about 1997. We have been working on this now for 3 years or so. So using, again, the CRISPR-Cas9 molecular scissors, we can knock out the IPC synthase in the model species Leishmania mexicana. Now, I won't go into detail here, but we lose all activity. There's no IPC produced in these parasites because they've lost the enzyme. So it's a real knockout chemically. And we do see the increase in ceramide as well. Remember, ceramide is a substrate. So you lose the enzyme, you will get an increase in the substrate. And then we this was work done with York, and we look at the knockouts in mice, and we also look at a line when we've added back the enzyme to the knockout, so it's complemented. So this line going up here quite steeply is the lesion being measured days post-infection of the parental line. This is our starting line, and the knockout is shown here. Now we do get a small lesion, but it never gets beyond about 1 millimeter. Now the add back does not recover. Now we don't know why, but we are looking into this. But I will show you something shortly that will be of interest. Now I should say also that a paper has come out in the last few months, the end of last year, that does this same experiment, but with conventional homologous recombination knockout. And they actually, in a different species, and they show that the knockout actually becomes hypervirulent in an animal model. So they're completely the reverse of what we're seeing. It's complicated. Frankly, it's a bit of a mess. On the other hand, now we're not here to talk about Trypanosoma cruzi, as much as Carlos would like to talk about Trypanosoma cruzi, because that's his favorite parasite. But very, very similar results in work we've done with Santuza Teixeira in Belo Horizonte. And we see this is the similar animal response, days post-infection. The wild type, we get hyperinfection. The knockout and the add back produce nothing. Okay, so and the animal's survival ends for the wild type, it just carries on further, both the knockout and the add back. So we're looking into this. This is now, we've done some further lipid analysis. This is now under resubmission to a journal. So one of the interesting things that I've only just seen really, because Santuza only shared this with me a couple of weeks ago, its recent data, is that those of you who know anything about Chagas disease will know it's in the heart. This is where it causes a lot of problems as a chronic disease. And whilst the knockout has a very low number of parasites in the mouse heart, the add back is quite high. But what we're lacking is the parental control here. Okay, so that's target based. I'm going to just, how am I doing for time? 10 minutes? 10 minutes, okay, good. Just I saw some yawns in the audience, so I don't want to hear you. Maybe people are asleep on Zoom, I don't know. I can't see them. So, okay, so that's yeast-based approach. I'm now going to talk about phenotypic approaches. This is the approach that's become most popular for looking for antiparasitics in general. So, whilst we have for all compounds, and this is out of date, so it's finished, this is the 10 years up to 2008. For all compounds, target-based sort of biochemical screening produced the most drugs that went into the market. Phenotypic, quite substantial, and they're substrate analogs of enzymes, small. But for first in class, this is what we call new chemical entities. So this is novelty, and your natural products, people largely, you're all interested in novelty in a chemical sense. Phenotypic screening, much more successful than getting new entities. Okay.
Can I ask a question?
No, yes, of course you can.
How many of these are structure based?
In 2008, probably 1 or 2. You tell me how many now?
7 days.
Yeah.
Good afternoon.
In the afternoon. Okay.
But there would be a target based. Structure based.
Yeah. In that, yeah. Because obviously you know that we haven't talked about that. So. Okay, so this phenotypic approach was taken again by GSK, didn't involve us, multiple people from different labs across the world involved in this. And basically, the entire GSK library, those 2 million compounds were screened against Leishmaniasis donovani in infected cells, T. cruzi infected cells, and Trypanosoma brucei, which are free in culture. Okay, sort of the nearest to pathogenic forms you can get in a high throughput manner. Now, that's the GSK library, and I'll talk about that and the success or not of that process tomorrow and Thursday. But now, the love of all your lives, maybe apart from your family, would be, of course, natural products. So I just want to make the point that in 2015, and this is the first Nobel Prize in parasitology for 100 years, more than 100 years, and ivermectin and artemisinin, respectively for nematode and, of course, malaria. The individuals involved in these, Campbell, Amura, and Tu, won the Nobel Prize. So natural products do make prizes if you're lucky, and work very hard, I should say. So natural products. So Patrick and I actually had a communication with this company called Oriogen, which is in somewhere south of Chicago in the US. So they're looking at this Oriobacidin A as an antifungal and some chemical modifications of it to improve its properties. So back to Toxoplasma. Now, one of the things about Toxoplasma, it has an encysted form. So probably at least half of the people in this room have cysts of Toxoplasma in their muscle and brain. I mean, that's just what happens. Unless you're a strict vegetarian, which some of you may be, then that's likely to have happened. So there's no treatment for chronic toxoplasmosis with the cysts in the brain and muscle tissue. This is a problem. It's associated with reactivation of infection on immune suppression. It was very problematic in the AIDS crisis, still remains problematic where AIDS exists. And also it is associated with mental health disorders as well. That's controversial, but chronic toxoplasmosis does seem to be associated with certain mental health problems. So interested in these particular compounds, that's Oriobacidin, that's a chemical derivative. And we were screening against an in vitro model of the cysts. And we can get hits, and this is one of the very few compounds that has activity against encysted toxoplasma. That doesn't mean it would clear the human brain of toxoplasma, but it does mean it at least can in vitro target cysts. So yeah, so that's how we do it, but it does not inhibit sphingolipid biosynthesis. I think you edited this, didn't you? So you asked us to change the title. As I remember. So there's our two natural products, anti-leishmanias. Okay, so I'm going to finish because I'll complement this on Thursday with just a short story about a project we have with a company called Hypha Discovery, which has a library of fungal extracts. And these are the kind of extracts that many of you are working with, which are mixed extracts. Okay, lots of different compounds extracted from, in this case, fungi. You may be looking at plants. So we screened these against Aezinicate mastigotes. We identified hits. We found one after lots of, and I'll show you this on Thursday, lots of iterative screening. And here it is. So we have Aezinicate mastigotes, and these are infected cells. So it maintains the same level of activity in infected cells as with free mastigotes. And it is more active than it is against the uninfected macrophage, but it still demonstrates a fairly high degree of toxicity. There it is. So it's this highly oxygenated and unsaturated molecule. Now, I haven't got time. I was going to ask you to have a think about why, but have a think about why that's a problem and why it may be toxic. Be now Thursday, and we can cover that then. It's novel, and I'll tell you this now because it may, influence some of your thinking in the next 48 hours. It's novel, but it's a very rare component of the mix that this was fractionated from. So it'd be very difficult to extract large amounts. but it's really the chemical nature of this I want you to think about. And then, even though we probably wouldn't want to take that forward for drug analysis, we did want to know what it did. So this is where mass spectrometry comes in, where we look at all the small molecules, all the small metabolites that are in the cell, in the Leishmania, when we add a compound. In this case, our natural products that we identified. Now, some things go up and some things go down. Now, the things that go up, we have a very large rise in L-cysteine. So that's an indication of oxygenation, okay? Oxidative stress, the production of cysteine from cysteine. And we have a very, very sharp decrease in trypanothione. Now, I'll talk about trypanothione very briefly, but it's key. There it is in the peroxidase system. And this is what the parasites, Leishmania and the trypanosomes, use to counter oxidative stress, which they face in all sorts of circumstances, including on drug assault. Okay, so it is essentially taking out that trypanothione, which is basically the parasite equivalent of glutathione. Okay? Very, very important. Okay, so all of this suggests oxidative stress. And then I want you to think again about the chemical composition of this, because these two groups here with saturation and the oxygenated areas really have the potential to act as Michael's acceptors, which means they can interact covalently with such things as trypanothione. Carlos.
One question, do you know the mode of action of the as an antifungal? Because
No, it's not antifungal, it's a fungal product. It may be antifungal, but it's from fungi.
I didn't really know the function.
No idea.
Okay.
It's a really, really minor component. Tiny, tiny quantities, very difficult to extract. So essentially, it's activity towards nucleophiles such as trypanothione and glutathione. So it's literally covalently attaching to these and removing them from the system. So it's going to be lethal. But we as humans ourselves rely on glutathione, so it's pretty lethal to us as well. And so just have a think about structures of natural compounds in general and compounds in general. Yeah, question.
Yeah, I'm thinking about this the microconcept, and I have worked with the SS department electronics, which have a lot of this concept, and they have been shown to have concept on the fire. Probably this is something maybe you can try and think about, because they have similar structures, infrastructures that.
I think that's something maybe we should talk about later. We're not really, because of the problems with extraction and the fact it was not that promising as something to develop, we haven't followed it through, but I'd be interested to talk about that later. So thank you. Okay, so I'll leave it there. There's a few things to think about before Bartira speaks next, but also before everybody gives their talks. Please continue to be interactive, interrupt. We can't just rely on Iqbal to ask all the questions. But just to emphasize, we have our team in Durham, and most important, well, as importantly, we have the international team, the interdisciplinary team that we work with across the world. And as you can see from the list, a large number of the individuals there are in this room today. So that's very important. And of course, their funding is very important as well. And I have to say, increasingly challenging, but we should talk about that separately. So thank you very much.
We have 2 minutes. Any pressing question? Yes, please.
Speak kindly loudly. This is my question is, what should somebody share when it comes to products. It's not only drugs or any product that you, and you do research on it. What can you share? Because some of the scientists only do presentations, they try to hide secrets. What should we share out there?
Well, for the benefit of our online audience, I'll just repeat the question. So the question is a very sensible, very interesting question, a very difficult one to answer. When it comes to identification of natural products, what information should we share? So it really comes down to the conflict between what I would call pure research and academia and impure finance and company expectation. So won't surprise you that I'm on the side of the pure academia, but I know that we all have to work somewhere in the middle. Okay, so I think the thing to do to give reassurance to your university to try and get a pattern as strong as possible and then share as widely as possible. I mean, that's the common route. It depends where you work, how good the university sort of legal structure you have. But genuinely, if you want to, there's no point doing science unless you share it, in my opinion, because otherwise, how are we going to develop this globe? But if you're worried about commercial interests, then intellectual property protection is the way to go. Good question, then.
Yes.
Question. So, for the presentation, you've been targeting like one key pathway and one specific enzyme, IPCS. So, I'm just wondering, like, can the parasites not adapt and probably use other pathways to, like, now overcome the trying to?
Yeah, that's another good question, because the question just for our online audience was to, if we target an enzyme such as the IPC synthase, is it not likely the parasite can adapt and avoid that inhibition? Of course, that's the principle of drug resistance as well. And we know in Leishmania, as we do in bacteria, and indeed in cancer, that drug resistance happens very, very quickly indeed. And many of the drugs that I showed on that list have very effective resistance pathways created then subsequently in comparison. So it's complex, but on a sort of biochemical level, it's entirely possible that, and I think that was one of the points I was trying to make with the knockout and the loss of unexpected genes and proteins. It's entirely possible, at least in a cell culture, which is not a very aggressive environment. It's not like being in a sandfly gut. It's a comfortable environment that the parasite can adapt because the genome is plastic and loose or even overexpress certain proteins to adapt. So I think we're only really starting to understand what the parasite can do genetically to overcome that. The third point I want to make there is that a whole class of drugs, the antifungals, azoles, which target sterol production in fungi, were proposed as anti-leishmanials because there's similarities between sterols in fungi and leishmanials. Indeed, they do work. But Leishmania can easily use the host cholesterol as opposed to its own ergosterol. So they were quickly disposed of. Even though some people still insist on working on them, I wouldn't touch it. So you have to be aware that there's an interface between the parasite and host as well.
Okay, so last question over there in the interest of time, and then you can perhaps discuss this during the. Yeah, sure, please. Please.
My question is hydrogen, and I'm wondering whether it's possible if to participate with the enzyme.
possible to, sorry, I didn't get the question.
So I'm asking whether it's possible to do virus screening or with this enzyme that we use for high screens.
So to do it as an enzyme assay.
So the virus screening to reduce the number of.
So which enzyme are we talking?
Virtually.
Yes.
But this enzyme,
The IPC synthase.
yeah.
So I think, tell me if I'm wrong, but I think the question was, can we do an in vitro assay?
No, in silico.
In silico, sorry, in silico. Well, I'm going to pass this over now to my esteemed colleague who does actually do in silico analysis, Professor M. Capola. You have to come here then.
Yeah.
Easy question. Is the answer? Just wait until 3 o'clock. Okay, I'll give the answer.
Broadly, the answer is no.
Okay, thank you very much. Thank you all for excellent presentation. Wonderful presentation to set the stage for colleagues. Now, I have the honor of inviting another wonderful scientist in this field. She is from Brazil, and now inviting Dr. Matera Rosie Schwartman, and she's going to talk about discovery and development of active compound influence leishmaniasis. She came all the way to the other side of the planet. So, Matera, over to you. Matera, yes.
Okay.
No problem. No problem. It's very okay. Just 1 minute before you start. Sorry. Just let me check something.
You want your water? Yeah.
Sorry. Okay. No?
It's not working anymore.
No,
It's just the point of it. I don't think that's.
Okay.
That's not one. So. Okay, hello again. And so my talk will be on discovery and development of active compounds against leishmaniasis. And as you see later, it will focus mainly on the natural product. Okay, so I'm glad that Paul has already talked about some of epidemiology of leishmaniasis. And just this is a different way of representing both cutaneous and visceral leishmaniasis together. So the darker countries are the most have been most prevalent cases of visceral leishmaniasis, like Brazil, Sudan, South Sudan, Ethiopia, and India and Bangladesh. And the others are cutaneous leishmaniasis, which is more widespread. And So, sorry, so Kenya here just to emphasize that you have both visceral and cutaneous in a medium scale of prevalence. No? So I'm not going to go again on the life cycle, but what I want to show is just that this parasite is an intercellular parasite, that the initial infection in the skin, macrophages, when the infection remains in the skin, you have cutaneous leishmaniasis, and when the infected cell migrate to deep organs like spleen, bone marrow, and liver, you get the visceral leishmaniasis. This is a very difficult disease to treat, mainly because it is an intercellular infection. So it's very difficult to make the drugs to reach inside the cell. One of the reasons, not the only one, but the main reason is because it's intercellular. And this timeline shows that there's not only the technical problem of being an intercellular parasite, far from the reach of drugs, but also because it is neglected disease that there was not much development in new drugs in the last century, because the industries that produce the drugs are not very interested in investing in disease of the poor. So all these drugs, trivalent antimonial that were later developed in beta-valent, less toxic, betaine, amphotericin B, pharmacin, and then the amphotericin B liposomal formulation in the 90s, and the teflozin was the last one. So all these drugs are repositioned from other diseases. None of them were discovered and developed for leishmaniasis. Okay, so today what we have are a combination of all drugs. We don't have a new drug on the market. And so that's why I mentioned earlier this morning that we need better drugs and new drugs for leishmaniasis to substitute this old ones that are still used, for more than 80 years, same drug, like the beta-valent antimonials here. And why? So these are the different forms of disease, the cutaneous leishmaniasis. If you can see the thing. In the battery splash.
Oh, okay, that's why that's why.
But as you can see, all treatments that are used today, they are given by injections, except for nifedipine. That's an oral drug that's being used mainly for visceral leishmaniasis, and it has been introduced in Brazil recently for treatment of cutaneous leishmaniasis. But still, we don't know how it will work. It has been not being adopted by the doctors yet, the oral treatment with nifedipine. And that can be used only for cutaneous leishmaniasis because the treatment of dogs with visceral leishmaniasis is dogs are already using nifedipine. And so we'll soon have resistant strains around the country, and it's possible that humans infected with Leishmania infantum that are resistant to nifedipine. So that's why it's not being given to humans, nifedipine, only for to treat visceral leishmaniasis, but only for cutaneous leishmaniasis. But it's perfectly done. And also the problem, another problem with these drugs, besides being very toxic, all of them have their own toxicity, is that they are given by multiple injections, which makes it very.
You want
Oh,
to
yes, please. So the multiple injections that makes the drug to accumulate and become more and more toxic. So there are about 30 injections per series of treatment. So that's quite a lot. And the main issue is localized cutaneous leishmaniasis that sometimes you have just one lesion. And even so, the patient has to go to all those injections for 1 month just because of one skin lesion. So that's unacceptable, no? We can have in Brazil some doctors prescribing intralesional injection with antimonials, not only in Brazil, but in other places, in Pakistan.
Also, but it creates nodules, it creates another structure in the lesion, so this injections.
Yeah, it works well, yeah, but you have to still keep giving repeated injections because the antimonium is very hydrophilic and is very quickly absorbed to the circulation. And there are some cases in Brazil that even if you give it intralesional injections, you can get toxic effects in the heart because it goes to the circulation. Okay. And so we are focusing on localized leishmaniasis, which is by far the most common form of the disease in the world. And for cutaneous leishmaniasis, more than 90%, and it's caused by at least 20 different species of the parasite. They're called cutaneous leishmaniasis. And in Brazil, we have these two species here, Leishmania brasiliensis and amazonensis, that cause this very dreadful form of disease, which is the mucosal and the diffuse leishmaniasis. So that's why it's important to treat the disease while it's still in the localized form, or it just transform and metastasize to other, to the face of the body. And so I have already mentioned the problem was systemic toxicity, that makes WHO and the NPI to emphasize that we need say oral and topical drugs for cutaneous leishmaniasis. And why is treatment of cutaneous leishmaniasis so difficult? It would be very, one would imagine just a cream would be good to treat, but that's not happening because as the lesion grows, you can have this ulcer here, and the borders of the lesion, which is where the parasites are in the border, gets thicker and thicker, the hypertrophy of the tissue. So the drugs cannot cross the top barrier, which is the stratum corneum, and it's very difficult to cross here, where you should get the drug to reach the parasites in the dermis. So that's and some of the drugs that are used to prepare creams for topical treatment, and they are either large drugs or they are not lipophilic. There's this one, the tercin B and pyromycin, that are the drugs that have been used to commercialize cream for Leishmaniasis treatment. And this, there seem to be, besides, well, this is the less hydrophilic of them, but it has also the highest molecular size, which makes it difficult to cross the skin barrier. And phenolmycin, on the other hand, it's not big molecule, but it's very hydrophilic. So none of them have produced really very good cream to treat the disease. So we still don't have any topical cream. So we need new drugs, and we can have, this is a scheme of the smuggling and development. The chemical, synthetic or natural products normally are assayed on parasites and cells for antiparasitic activity and cytotoxicity. And the good ones, the more selective one, go for animal studies, where they are tested particularly for efficacy, and the good candidates may go for formulation and clinical trials. And then after passing the clinical trials, trial, we could go to the market. But as Paul said, we don't have any new drug that passes these phase 2 clinical trials here. And so not all laboratories have the access to animal studies, particularly in Europe. It's very difficult. And also because we have to keep the animal numbers to a minimum. No, this is for professional issues. No, we try to use only the drugs that have really very good indication of the passive in vitro tests. And another approach is then the more rational approaches, which is that you can have your natural product, and the active one you can have the structure, and also synthesized analogs, that may have better activities, and synthesize and then test again, and that's another way to to move ahead with an interesting lead compound. What else you can do if you have the enzyme package, the protein, you can do docking analysis and then design new leads that will bind to the package and synthesize them and then test for in vitro studies and so on. Well, this is, like I said, a more rational approach, but plants and other natural products also have a very good possibility that we have a good anti-Leishmanian activity. And some compounds, as Paul showed, they are very, very complex. So complex that you would not be possible to synthesize in the laboratory. So some chemical natural compounds that are from plant may possibly go straight to tests, not synthesis. And that's some of them have given very good results. And we have now what I'm going to talk about from now on is about what we have done on plant compounds and reached at these stages here to formulation. So we tested about 100 extracts from Brazilian plants We did some screenings on Leishmaniasis in vitro. And then we also in vivo in animals. And we found some chalcones, especially a plant called Peter, which is very rich on these chalcones. And target identification, nanoformulation for oral and topical delivery. So that's what I'm going to talk about. And this is the plant, we identified by the chloromethane extract this natural chalcone, which is quite simple. We did some synthesis of analogs of this, and we got this new natural chalcone that we call the chalcone CHH. And this is a very lipophilic compound, has a very good anti-Leishmanial activity, good selectivity. And we tested information in mice. They were infected with Leishmaniasis, and then they were treated with 8 injections in the ear. And we compared the chalcone, giving them a dose much lower than pentostan, and we found that a very good reduction in lesion size growth. No? So we have this promising prototype drug that so after having this in vitro and in vivo results, we said, well, we have to identify the target, and we know that chalcones are very ubiquitous compounds. They bind to different proteins, but we decided that we could go forward, and that was about exactly 10 years ago that I met Patrick Steele, and he was also interested in in fact, he was a chemical biologist and a parasitologist, so we put together our interest in defining the target of an anti-Leishmanian drug. And that's what we decided to do to get to the target. And in the mean of this studies, we got also the collaboration of Paul Denny and people. So we proposed to start with the synthesis of that was what Patrick was working on, fluorescence probes, and then to find if they could bind to parasite proteins. And then after that, with the gel, SDS gel, get the band for protein identification by mass spectrum. Then to see if the chalcone could physically bind to the common protein, to the docking assays, and then look out parasites, to see if it was the important targets that we had identified. To start with, we synthesize this new chalcone, which is also a nitric chalcone, but it is more easily synthesized and purified than the original chalcone CHH, and also as active as the original chalcone. Okay, just because it was more simpler to purify than the synthesis of this previous one. So this one was synthesized by our student in Durham University, and to serve as a probe, where it was this alkyne binding here to the probe. It was a trifunctional probe, actually, that we could bind the rhodamine tag to make it fluorescent. Okay. So this binding was by the quick reaction, yes, a series of reactions. And we had also to find if this probe could actually bind to parasite proteins. So we coated the tomato probe with the probe and added the rhodamine, and then after the reaction, we could see the focal microscope that yes, the probe could get inside the parasite and bind to the parasite protein. That was the initial demonstration. And after that, we had to identify which protein the chocolate was recognizing in the parasite. And we did that by culturing the parasites, lysing them, and then binding with the rhodamine. So we had all of the protein here that was bound to the chocolate was fluorescent. After STS page chromatography, we could see the fluorescent band here. So the band was cut and then analyzed by mass spectrometry, and we found that it was the cytosolic redoxing peroxidase. It's an enzyme that also participates in the same redox mechanism of the parasite. So this was it's a main fluorescent band here. So that means that this is the most important protein that was recognized by the chocolate, fluorescent chocolate. So this enzyme, the parasite enzyme, which redoxing peroxidase, is uses family of antioxidant enzyme, important for parasites to get rid of toxic oxygen radicals produced, for example, by the macrophage, important for effectivity and survival, and most important, it is highly conserved into chromosomes. So this enzyme may be what is recognized into chromosomal fluency and African chromosomes. And we had actually demonstrated before that the chocolate also kill those parasites. So it's possible that it's acting on the same enzymes, no? They have a common target. After that, we did that was worked on with Paul Manny, our student Douglas Scribani, went to the room and to produce the recombinant protein, this enzyme. And we can see it was after producing the recombinant peroxidase, it was set on thermal stability, where the chocolate was infiltrated with the recombinant protein, and then you could see the temperature, showing that it was really binding there, and also in MS that when the protein was incubated with the chocolate, you could have a change here in the pattern. And also the docking to find that really the protein was binding, sorry, the chocolate was binding to the protein. So after that, gene deletion, the parasites were deleted by CRISPR-Cas9, and these single deleted parasites were not affected by their growth. They were growing almost as normal as the white type parasites. The double deleted one was less able to multiply in vitro, to grow in vitro. They don't look very well, but it was enough to test the chocolate with these parasites, and actually found that we see there's, but the knockout parasites were less susceptible to chocolate. So That gave an indication that this peroxidase was an important target. We can tell that it's the only one, but it is the most important one. So that was published. Many people participated in this work. That's Douglas, the student. And so after identifying the target, which is very good to understand how the drug works, and also to predict what sort of drug combination we can use, so that we use, for example, treatment with a chocolate and another drug that acts not on the same target, to have an additive effect, you know? And also maybe use the for molecular modeling, for example, in the new analogs for the target. So, and that's that stage, and we moved to formulation and to make the drug more effective. We used some formulations that made do a lot of improvement in the effect of the drug by increasing the stability, the absorption. If we wanted some drug that could be absorbed by the skin, targeting to the macrophages, and we can use lower doses. That by using lower doses, we can have a reduction in overall costs of the drug. No? Because some drugs are can become very expensive. Just to think that it's helpful. The less you use of it, the less will cost the treatment. And also, the important that you have a reduced side effects because of the increased targeting to the target cell. So just to mention the targets that we would find when you envisage nanoformulation for better absorption through the skin, for topical treatment, or for the oral treatment that you have to pass the acidic environment of the stomach, the digestive juices in the intestine. Now they have to cross the mucosa and escape liver metabolism. So it's very difficult not to get one drug to reach the plasmids in the skin without suffering and some of these attacks during the way after the oral ingestion. And also in the end, eventually you have also to cross the macrophage barrier and the parvolysosomal membrane barrier as well. So there are many types of nanosystems that we have used in our lab, and we use this chalcone HH as a drug prototype. This is a if you look at this drug in the microscope, you see very huge crystals, and that makes it very difficult to be absorbed to the skin or to the by intestine. So you can use these nanosystems here to make them smaller and be absorbed in more easy way. And I'll talk only about these two here. I actually treat nanocrystals, and polymeric microparticles, and polymeric nanoparticles capsules. Okay, we can have use all of this, but they're only the best ones I'll show you. So the nanocrystals are very interesting because we can use the dry milling and the wet milling. You can transform in nanocrystals here, you can see, and in this way you can improve the the suspension of the drug. You can see here, this is the not created on, and the nanomat. You can, it's much more stable in water than the big crystals, and also the solubility. And we prepare this suspension here for oral delivery in mice infected with Leishmaniasis. And actually, this is in plant line is the untreated animals, the lesion animals giving the big crystal of chalk in blue, and the nanocrystals in red. So the nanocrystals become much more effective than the big crystals. And here, similar to glucantine injections given intravenous. So this is a way that you can improve. It's not difficult to make the meal in the lab. So of course, you have to have the equipment to do that. But it's some drugs that are very lipophilic that you would just them aside because they are not absorbed. You can easily do some milling to make it work. And so another one is the polymeric nanocapsules for topical application. And we prepared this nanocapsule made with polymeric caprolactone. And the inner core of the nanocapsule is oily, where the chalcone can be solubilized very easily. So it measures 120 to 20 nanometers, no, the diameter. And we applied to the shaped neuron of the mice. No? So the idea was that it would be either absorbed through the stratum corneum or be deposited into the hair follicle, and from there the chalcone would be released within the skin. And what we did was we prepared some fluorescent nanocapsules labeled with rhodamine, and these were loaded with the chalcones. And here you can see infected macrophages in the bottom. This is just light microscopy. And then the fluorescent microscopy is showing in green the parasites, GFP parasites, no? And the green, the orange, sorry, the red is the nanocapsules. So they are localized inside the macrophages. And in vivo, we applied on the top of the skin, no? And followed the disappearance of the fluorescence with time. And after a few hours, we could see that all nanocapsules had been absorbed. That was not just because of the fading of the fluorescence, because we applied that on a plastic surface and it remained fluorescent to all the time that we followed the experiment. So this demonstrated that these nanocapsules could be absorbed to the skin in this way. Okay? And we compared the application in mice with the chalcone in cream pharmaceutical formulation. These the nanocapsule alone. They had some effects on itself. It's very strange because they may have been absorbed and gone to the macrophage, and the particle alone could have some healing effect, activation of the macrophage to kill the parasites. That's what we guessed. And this in yellow here, the nanoformulation showing that it's much better than when the drug was applied in a drip form. Okay. So this one we had so far, this one for oral delivery, this one for topical delivery. Another way of delivering the drug for pain leishmaniasis is to bypass the skin barrier by giving it intralesionally with a syringe and injection. So we designed this polymeric microparticles loaded with the charcoal. So the idea was to reduce the multiple doses that are used with the leishmaniasis, for example, to just a single dose, for controlled release in this type of infection. So one single dose would make the same job as the repeated doses. And then what we are now doing for the clinical trial is injecting in different parts of the lesion. The patient must have not more than 4 lesions, measuring 3 centimeters each, and giving it intralesionally. After weeks, the polymer dissolves and release the drug. We can have reduction after a single dose. And we did that the clinical trial. Oh, sorry. Just to show you how it works, the big crystals are dissolved in the chloromethane and water to be formulated in the microparticles. You can see here, it was not homogeneous, but had a very good size distribution. And they measured, they were bigger than the previous nanoparticle. This was measuring around 6 micrometers. So the preclinical study, the animals were infected in the ear and then given 1 single dose after 30 days of infection, or 3 doses. We compared a single dose with 3 doses, and they were killed 3 months later. And you can see here the animals that received 1 single dose here. We have a much reduced lesion size, much reduced parasite load, and the one that received 3 doses of the free drug. And 3 doses would really almost get rid of the parasites. So we demonstrated that 1 single dose can have a very good effect. It's very difficult to get rid completely of the parasites in the lesion, but if you reduce it to a level that the immune system can keep it to a low level, that's good enough. Okay, that's what happened with most of the treatments that we have in leishmaniasis. So we so far have that, and we, as I said, we are doing points for clinical trials, not with this formulation here, and then tell you why later on, because it's very difficult to get approved first drug that we have never been used in humans before, and this charcoal, and also with the formulation is another step that you have to show the safety before you go for the clinical trial. So we are now testing another form of producing these microparticles that is more reproducible and more easily approved by the regulatory agency. We use the microfluidics that is with the fabrication of monodisperse macroparticles and nanoparticles. It's very easy to scale up. And this system can also be used for organ ownership or body ownership, which is good to test new drugs without the use of animals. And here we show the conventional way to prepare the microparticles that you have, it's not working, but you have a wide dispersion in size. And when you use microfluidics, you have a very, very sharp dispersion of size. So this is very good for reproduction and acceptance as for clinical trials. And so we are now in the process of fabricating these sheets in the lab. By this is a quite complex way of preparing them, many variables. So we've been here in this stage for quite about 6 or 8 months. And until we get a very good way to prepare and very reducible way to prepare these chips. And the good thing about microfluidics is that unlike the batch way of preparing the microparticles, that you have a very big variation in size, and you do one day, the next day you get completely different particles. Is that you can do scale up for industrial process. You can stack the devices one to another, and so by the industry you can produce very large amounts of microparticles in this way. I'm not going to go in detail here because it's short of time. So the last slide. Almost here, as I said, for the clinical trial, we are now using amphotericin B loaded microparticles just to open the way. We are now just going for the patients to selecting them to test. And after that, we believe that once it is established this new way of treating cutaneous leishmaniasis, we can introduce other new formulations with new drugs like the chalcone, for example, perhaps with a combination with amphotericin B, and because this one has worked very well in the clinical, and I didn't show here. But so I just try to provide you what we did is screening of plant extracts to target identification, formulation, and delivery. You know, I think the natural products are very promising, although we chose a very simple molecule, that is chalcone, so is not difficult to synthesize, no, different from complex products like amphotericin B, for example. That's very difficult to synthesize. It's possible, yes, to synthesize, but it's not trivial. So that's what I have to show you. And last, oops, and back. Sorry. And the collaborations. Yeah.
Was there?
Well, but I wanted to thank all people that collaborated and here present Patrick Steele, which was the one that created the first steps of the work that we did together. And then people, and yeah, thank you very much for the audience.
Thank you. So let me open the session for questions. Yes, please. Yeah, the second U, third U, fourth.
So can you please speak louder, please?
Yes, yes.
Nice presentation. I want to ask, the choice of Jumbo, because for the Brazilian snowflakes, there are thousands or hundreds of secondary developments. So your choice of Jumbo is guided by the local activity or the simplicity of structure.
Oh, we didn't know the structure before. We tested the extract. And after getting an active extract, then we identified the active compound. And even have with C41.
Then I elaborate this question.
Yes.
How was initially this plant was selected? What was the strategy for the?
For selecting.
Yes.
Oh, yeah, well, these were the people, the natural product people at the university that through taxonomy.
No, I mean, was a phenotypic screening done for many extracts?
Yeah, they had they knew that this plant species were rich in some compounds, but actually we were looking for safety terpenes, and we found some active ones, but the chalk on was sure to be better, so that's why we looked at it.
The first question that Dr. is for the planters. When you try the crude extracts, and if the crude extract is active, why not directly go to drug development from her?
Culture this the plant?
No, additionally, the one that plant that already there, not development, active, where it is active at crude stage. Why you are not going to directly develop a drug from herb itself as a crude extract?
Oh,
And
why
additionally,
didn't I use the natural charcoal that was extracted? Did I move to something?
Plant extract, because they're asking that one practical stuff which is more in the context of developing a world rather than purifying a compound. Why can't we develop some formulation, topical application from the extract?
Oh, directly from the extract, because this plant, the charcoals are extracted from the inflorescences, and it is produced only during the summer. And the student used to collect the inflorescences, inflorescences of the plant and make a batch. But then our summer is in January and February. And then when it was November, we got all the powder we had used, and we had to wait again. So it was a matter of the purification was not, the yield was very low. So that's why we synthesize it.
You please. I mean, during the lunchtime, you miss a late lunch, you have a choice, we can continue the discussion. And continue with the third speaker, is this okay? No? You want to go for lunch? Then two more questions. Over to you.
Okay, thank you for the presentation. I think that you communicated communication of product tested in animals. Then finally, when you wanted to scale it up, when you wanted to do the clinical trial, you used microfluidics. So if I got you correctly. So my question is, for someone doing similar work, will you maybe advise them then to, to skip the manipulation bit and just use microfluidics? Because like you correctly said, it's a challenge convincing people about the safety of the nanoformulated. Would you advise someone to just now that you've gone through the truth?
Well, each new drug you have to prove the bioavailability, pharmacokinetics, the safety, many different ways to make it alteration in neighborhoods. So it's very time consuming. We have some safety, preclinical assay done, but every time our health regulatory agency, so-called Anvisa, is history. So every time they ask for different thing and they were never happy. So we decided to move to comparison B, which is already approved. And just the formulation, when you have a formulation of a compound, they treated it as a new drug. It's a new drug. So, but it was much less steps than if it was a new drug. So that's why we want to establish that within comparison B formulation, and so because nobody has used before, the control release microparticles being for a single dose. After that, I think it will be, after we have gone through all the steps, we may know better how to improve the new drug.
So someone was in between here. You. So I can move it to you, or you can stand there.
Thank you for the presentation. So from the presentation, I think the future looks great on the Leishmaniasis discovery. So I have a question regarding the toxicity and the drug administration. So from the oral administration, so are there cases that have probably been reported on maybe alteration of the GIT? And the other question is about the dosage. So you say using the nanoparticle is more effective, which is probably low in terms of dosage as compared to high function. So what is the explanation that
The nano-formulated drug is more effective than the free drug. Is that what you're asking? Why would that be? Yes, that's a good question, because you can think that, well, you still have the nanoparticle to cross the intestinal barrier, which is, but we have in the intestine some cells called the M cells that are phagocytic, and so that's the way, and this polymeric particle bind very well to them, and that's the way perhaps that they are catched by the epithelial cells better than the drug itself. We have used a novel formulation that makes the crystals or the free drugs more in suspension, but it appears that the big crystals prevented the uptake by the so that's why we think that the crystals are too big to be catched by the M cells, unlike the nanoparticles. That's the way.
Thank you very much, Natara.
Thank you.
Thank you everyone for very interactive, listening everything about very exciting work being conducted. These two lectures were really of very high quality, in which you have learned about various stages in drug identification, the whole story of the development of a drug candidate. So exciting work that was presented, and I would really invite you to interact with the speakers during the lunchtime and in the evening time. Also, thank you very much, everyone.
So
We get the lunch or not?
Yes, yes. So,
It was, right?
so where the students, van was parked, okay? Will be guided, and then there's the bus, the two buses are there. They will take us to Toronto. And then we get us back and here. Okay? So we go down to the A44. Some of us, you are a veteran, you can guide. Yes, you know, we have Can you come in close, please? Yes. Just back here, place on the yes. Yes,
It's very important.
Yeah. Closer to the. Yeah, but we can share a lot of. Yeah. This young lady is very responsible. Yeah, thank you so much. Now we go. Good. What are you on?
Ama mnaenda unabaki? Umeenda lunch? Oh, inabidi tufanye lunch yangu sasa. Ndio kisa ngapi? Sio, wewe hapa kwa
I think I said it myself.
So good afternoon, our colleagues online. I'm sorry for starting late. This because as we went to our lunch place, found a very heavy traffic, which as a result, we were unable to start our program. We sincerely apologize, but we will start in the next maximum 15 minutes. Welcome and thank you for your questions.
Data is our session chair.
Okay, good.
Okay, so. Can I go someplace like?
Oh, okay. No problem. So the being is one. We go up to the 15th drop.
Okay, I think by the time people come,
No problem.
Party is supposed to be your reception. We should just start.
Not much. I should have finished.
Okay,
I should have finished by now.
thank you.
So thank you for your time. Down now. We did an hour waiting for you.
I thought you finished, that's why I can have stuff.
And just go to the bathroom. No, just go to the bathroom. Okay, that's fine.
Easier than walking.
Walking's very nice.
But yeah, quicker, because there was no, or little, I say no.
You'll be right, at the same time.
Yeah. We left considerably out. Dustbin anyway. Let's get mad already.
Myself and
It don't feel you have to speak for 2 hours.
I've always gone for it so long, so I've got no attention time.
Oh. You'll need to, put your, this is mine, but the other one's not working. Yeah.
It's okay, I might walk until keep myself
To the hotel.
where I'm actually.
Oh.
I don't have to stand.
Well, you do, because
It's up for the mess of.
yeah, that's, actually, no,
I know you're very presidential.
I know nothing. It's not something I'm in control with. I don't really think someone is in control of the complicated situation.
If we have one here at in a second, summer is certainly not in control. Well, the was which possible going back.
Well, yes, I'll be fine. I need to see it.
Actually, got some work.
Oh, all right. So you walk.
You're here.
So it's very easy. But I did yesterday pay 647 quid for those. That's it.
Makes me feel good.
Whenever it is open.
So, MP. Solomon is not around. So could you kindly help us to just share the slide?
Yes. Okay, how are you going to broadcast yours?
Yeah.
You need to put it on my laptop. We're gonna move.
I
Just wanting to connect the music. Take a screen.
I'm going to do starting.
I've done my work.
What can I say about what can I say about you? Yes, We should stop sharing this one first. Where is your first? This is not sharing the first one and then share this. Without doing this. We should share. Okay, but if
Oh, yeah.
you have the point.
Andrew, do you want me to ask different questions?
You can answer it, but if you want. You have to get people from our. It's not quite like that.
You change the size.
No, this one. Sure, fine.
Shall we start?
Online.
Where is the frontline people?
Yeah, this is amazing. Siesta time? Yeah.
Okay, good afternoon. I have great pleasure to introduce Professor Patrick Steele from the Chemical Biology Institute of University of Durham in UK. And Patrick is chemist.
I'm an organic chemist.
Organic chemist and also more recently parasitologist as well. And a great friend also scientist. Okay, Patrick.
Thanks very much, Dr. I like to wander around. I like to walk and talk, so I'll hold this. You can't hear me. Stop. If you don't understand me, stop me. You've got a question? Stop me. Okay. I've got quite a lot of material. I'll go through some of it very quickly. I'm going to try and highlight principles rather than anything greatly significant. Maybe I'll start. But not quite the correct type on the program, but that's what my group has been doing for a number of years, mainly with Paul, Bartira, and other people. I'm a chemist, I'm an organic chemist. I discovered today that a professor who rides a bicycle and doesn't have a smart car is somewhat crazy. So my mission is